Na plus K plus pump or Na plus K plus ATPase, because it uses ATP as a source of energy and transports sodium and potassium against concentration gradient. So sodium potassium pump is a very ideal example for primary active transport. Let us try to understand the functioning of primary active transport here. A sodium potassium pump is one of the very interesting pump which is widely studied in medicine. The sodium potassium pump is made up of three subunits: the alpha, beta, and gamma subunits. The alpha subunit, it has three subunits of proteins: the alpha subunit, the beta subunit, and also we have a gamma subunit. The alpha subunit perform the major role of transportation, whereas beta and gamma, they have major role in anchoring the protein to the membrane or to regulate the function of the alpha subunit. How does this pump works? This pump, it works by transporting the sodium against the concentration gradient from inside to outside the cell. So this pump, it transports 3 sodium. How many? 3 sodium from ICF to ECF, that is from lower concentration to higher concentration. So 3 sodium are transported from lower concentration to higher concentration, that is from ICF to ECF. It transports 2 potassium ions. It transports 2 potassium ions from ECF to ICF. 2 potassium ion from ECF to ICF, again from lower concentration to higher concentration. At this juncture, I would like to add one point. Always remember the distribution of the ions and molecules we have studied in our first chapter. Always remember the distribution of ions and molecules which we have studied in our first chapter. Remember, sodium is always more outside and potassium is always more inside. So, in diffusion, it moves from higher to lower. But in active transport, it is moving from lower to higher. So, sodium is more outside. But still, we are transporting sodium from inside to outside. That is from lower to higher. Potassium is more inside, but still we are transporting potassium from outside to inside. So remember, the concentration gradient has not changed. The concentration gradient has not changed. Sodium is more outside and potassium is more inside. Still we are transporting sodium from inside to outside and potassium from outside to inside. Because this is an active transport, this is an active process where they are moving against the gradient. It uses ATP as a source of energy. So ATP is used as a source of energy. Now there is one very interesting question here. This pump, it transports 3 sodium out, 2 potassium in, using 1 ATP as a source of energy. So a question which is always asked, that a sodium potassium pump has how many binding sites? How many binding sites? And remember, whenever you have such question, the answers are quite tricky. And for sodium potassium pump, the binding sites, if you have a question that the sodium potassium pump has how many binding sites, remember the sodium potassium pump has 5 binding sites. It has 5 binding sites. Let us understand which are those 5 binding sites. So if we say a sodium potassium pump, so it has sodium binding site on the interior face. We have 3 sodium binding sites on the interior face. 3 sodium binds onto the interior face. 2 potassium binds onto the exterior face. So these are the 2 binding sites, 1 and 2. We have 1 ATP binding site on the interior face. So ATP binds onto the interior face. And we have one enzyme for ATPase binding site. So ATPase binding site. So we have 1, 2 and 3. I'll repeat here. 1, 2 and 3 binding sites on the inner surface. 2 potassium binding site and one OA-bane binding site. It's called as OA-bane binding site, a site where the drug digoxin digitalis binds. Digoxin digitalis is one of the group of drug which inhibits sodium potassium pump. So OA-bane binding site. So in this way, this pump has 3 binding sites on the inner surface, 2 binding sites on the outer surface. In all, it has 5 binding sites. This is one of the very important question you are supposed to remember. Let us study the function of this pump. How does it work? So this pump, it occurs in various states. A state where it is, you can say, the uh, stage 1, stage 2, or you can say configuration 1 and configuration 2, and so on. In the resting state or in the configuration 1, this pump is binding to the sodium ions. It binds to the sodium ions onto the interior face. It binds to the sodium ions onto the interior face. I'm just drawing one of the, just a representative diagram. It's not a very accurate one, but just to give an idea. So initially it binds to 3 sodium ions onto the interior face. So the 3 sodium ions bind onto the interior face, and one ATP binds onto the interior face. One ATP binds onto the interior face. Now when this occurs, the sodium ions have bind and the ATP have also bind to it. The ATPase enzyme gets activated, and this ATPase enzyme, it hydrolyzes ATP to break into ADP and inorganic phosphate. ADP and inorganic phosphate. This causes the release of large amount of energy, because once ATP is broken down, energy is released. This energy from the ATP is utilized by this transport protein, and it changes its structure. It changes structural conformation, and by changing its structure, this transporter, it goes from its initial state to the state 2, and in that state now it is open from outside but closed from inside. Open from outside but closed from inside. So it will be something like this, where it is now open from outside but closed from inside. So what happens now? The sodium ions which have been binding over the inner surface, now they are exposed to outer surface, and these sodium ions, they are released, or they are being pumped out of the cell. They are being pumped, or they are being thrown out of the cell. At the same time, because the pump is in excitatory state, it becomes prone to binding for the potassium ion. So two potassium ion binds to the outer face, and the moment potassium ion binds to the outer face, the pump comes back to its configuration of the initial state. That is, again, it becomes closed from outside and open from inside. Closed from outside and open from inside. And in this manner, the potassium ions are taken back inside the cell. The potassium ions are taken back inside the cell. So the pump, it takes 3 sodium out, but in exchange it takes 2 potassium inside the cell. 2 potassium inside the cell. So now the potassium ions, they are released into the interior of the cell, and hence the potassium ion is brought from outside to inside, whereas sodium ion is brought from inside to outside. So in this way it has got various configurations. Initially it is in excitatory state, or you can say in the stage 1, where 3 sodium binds and 1 ATP binds. Then when ATP binds, the ATPase breaks down, hydrolyzes ATP, energy is released. Using this energy, it pumps 3 sodium out. The moment it pumps 3 sodium out, it becomes quite unstable. It becomes quite a state where it can bind to potassium ion. So the potassium ion binds from the outer surface, the pump changes structure and brings the potassium ion into the inside the cell. So the potassium is brought inside the cell from lower to higher concentration. And all this process is done by using ATP as a source of energy. By doing this function, this pump serves many important functions. Let us list those functions. Before going there, I remember many students, they always ask, sir, can we remember this movement of sodium and potassium ion with this pump in a very simpler way? So yeah, there was a mnemonic. There is a mnemonic, and the mnemonic is called as Nokia. A mnemonic known as Nokia, where it can be used as sodium moving out, potassium moving in, using one ATP as a source of energy. I repeat, Nokia is one of the best mnemonic to remember sodium potassium pump. Sodium moving out, NO, potassium moving in, Ki, and 1A, that is ATP, is used as a source of energy. So in this manner you can remember the actual functioning of this pump. Now by doing this sodium potassium movement against the gradient, what purpose, what function does this pump serves? Very, very important function. Let us list those important functions of sodium potassium pump. Number one, the sodium potassium pump, it maintains the concentration gradient of sodium and potassium ion across the membrane. It maintains the concentration gradient. It maintains the concentration gradient of sodium and potassium ion. How? Let us assume that this pump does not exist. Just assume that this pump does not exist. So what will happen? Suppose if this pump does not exist, we will only have the diffusion possible for sodium and potassium ion. So if there is only diffusion is one of the thing which is available, sodium will keep on diffusing from outside to inside, and potassium will keep on diffusing from inside to outside, right? If there is only diffusion, the sodium will move from higher to lower and potassium will move from higher to lower. If this will keep on happening, a point will reach where they will reach a equilibrium, or a point will reach where they will no more move because all the gradient is disturbed. And we don't want this to happen, because if these things happen, then this will be very difficult for the cell to survive. The cell will only survive if we have more sodium out and more potassium in. So what happens whenever sodium diffuses? Whenever sodium diffuses from outside to inside, I repeat, sodium is going to diffuse. You can't stop it. Whenever sodium diffuses from outside to inside, this pump, it throws sodium out again. जैसे ही सोडियम बाहर से अंदर आता है डिफ्यूज होके, ये पंप सोडियम को फिर से बाहर पुश कर देता है. So sodium, if diffuses from outside to inside, this pump diffuses. This pump, it pushes the sodium from inside to outside again. And the moment potassium diffuses from inside cell to outside the cell, the pump brings the potassium back inside the cell. So in this way, this pump, it maintains the sodium concentration more outside the cell, and this pump, it maintains the potassium concentration more inside the cell. So it maintains the sodium and potassium gradient of the across the cell membrane. So it maintains the concentration gradient of the sodium and potassium ions. Remember, it maintains more sodium outside and more potassium inside. Remember the Nokia, sodium out, potassium in. So this pump maintains sodium and potassium concentration gradient across the cell membrane. Second function, it prevents the cell from osmolysis. It prevents the cell from osmolysis. How? It prevents the cell from osmolysis. A very interesting one. How does this pump prevent the cell from osmolysis? See, the sodium ion, as we have discussed in our previous session, sodium ion is hydrophilic. It loves water molecules. So what happens? The cell, see, the cell is surrounded by ECF fluid, and the ECF is containing a higher concentration of sodium ions. So the diffusion, as I told you, the diffusion is a thing which we cannot prevent. So the diffusion keeps on happening. The sodium keeps on diffusing from outside to inside. The sodium keeps on diffusing from outside to inside. You cannot prevent the diffusion. So when sodium diffuses, water molecules also diffuses along with sodium ion. The water molecules, they also diffuse along with sodium ion. So let me just draw some of the few water molecules for our understanding. So along with sodium, water also diffuses inside the cell. Now, suppose if you don't prevent this from happening, sodium will keep on diffusing and water will keep on diffusing. Ultimately, what will happen, that the cell will swell. The cell will start swelling up. The cell will swell up, and if you further does not prevent it, a condition might reach where the cell might burst off. The cell might burst off, called as osmolysis. Because of collection of water keep on moving inside, the cell might burst off. We don't want this to happen. So the sodium potassium pump help us to achieve this. The moment sodium moves inside by diffusion, and water also moves inside along with sodium, the sodium potassium pump, that is our Na plus K plus pump, it throws the sodium out again. So sodium is thrown out. Sodium is pumped out. And because you pump out the sodium, the water also moves out. The water also moves out along with sodium. Water also moves out along with sodium. And in this manner, the cell comes back to its normal size. So this pump, it works day in, day out, 24/7. And it prevents the cell from osmolysis. One of the application example, let us have one of the example. Say bed sores. One of the cause of bed sores is because of the lack of activity of the pump. What happens? Bed sores are the sores, or you can say they are the wounds which are seen in the patients who are chronically ill, or they are bedridden, chronically bedridden. So you can see their back develops some of the bed sores. What happens, because the person is lying down for a considerable longer period of time, say a chronic paralysis or a comatose patient, certain part of their back have more compression area as compared to other areas. So the areas which are more compressed, those areas of the skin does not receive blood flow, because blood vessels are also compressed. And because blood vessels are compressed, that area of the skin does not receive energy for ATP. And because ATP is not available, the pump stops working. And because pump stops working, the sodium diffusion keeps on happening, but the sodium removal does not occur. And those cells, they swell and they start going for lysis, necrosis and lysis. And hence, there is a wound which develops in that area. As a debris of cell death is happening over there, and that development causes pus formation. So to prevent this, now there are very modernized bed which dissipates the pressure equally, and there is no high pressure points which are created. But the point is that if the sodium potassium pump does not work, the cell will go under osmolysis. So two important functions we have studied so far. Number one, it maintains the concentration gradient, and number two, it prevents the cell from osmolysis. Number three, which is the third function here. Third function. The sodium potassium pump, it pumps 3 sodium out. How many? 3 sodium are moving out. Means 3 positive charges are going out of the cell. 3 positive charges are going out of the cell. At the same time, this sodium potassium pump, it brings 2 potassium inside the cell. So 2 positive charges are brought inside the cell. Because sodium carries a positive volt and potassium also carries a positive voltage. When 3 sodium goes out, 3 positive charges are going out. When potassium comes in, 2 positive charges coming inside. Means in overall this exchange, we are giving out more positive ions, we are taking in less positive ions. So if there is less positive coming inside, it means we are gaining one negative charge. We are gaining one negative charge. Means 3 positive going out, 2 positive coming in, loss of 1 positive in the interior, means gain of 1 negative charge in the interior. So whenever this pump works, it develops a negative voltage inside the cell. Whenever this pump works, it develops a negative voltage inside the cell. Remember, 3 sodium going out, 2 potassium coming in, means 3 positive going out, 2 positive coming in. Means there is loss of 1 positive coming inside. And when there is a loss of 1 positive coming inside, means there is a gain of 1 negative inside. And that causes the development of negative voltage. So this pump, it develops the negative voltage. So this pump, because it develops a negative voltage inside the cell, this pump is also called as an electrogenic pump. The other name for this pump is known as electrogenic pump because it develops a negative voltage inside the cell. Electrogenic pump. And in this way, this pump contributes in a very important manner to the development of RMP, that is resting membrane potential. So this pump contributes in the development of resting membrane potential, which we'll be studying in our other sessions of nerve muscle physiology and general physiology. So resting membrane potential is also because of the sodium potassium pump. So these are the 3 important functions. Let us just compile what we have studied so far. We have studied primary radio transport with an example of sodium potassium pump. The sodium potassium pump, it pumps 3 sodium out, 2 potassium in using 1 ATP as a source of energy, and this transportation occurs from lower concentration to higher concentration, that is against the concentration gradient. Sodium potassium pump has 5 binding sites. It has 5 binding sites and it has 3 subunits of the proteins. The major subunit is alpha subunit, which performs the action of transportation. The sodium potassium pump has 3 important contributions to the cell. Number 1, it maintains the sodium and potassium gradient across the cell, means concentration gradient of sodium and potassium ion is maintained. It prevents the cell from osmolysis. And the last one is it generates a negative voltage inside the cell. So these are the 3 important features of the sodium potassium pump. Other examples, we'll just write them down. Apart from sodium potassium pump, we have calcium pump, which works in muscle cells and other tissues of the body. We have hydrogen pump, which is commonly seen in the gastrointestinal tract, in the stomach for HCl secretion. We have another pumps also, but majorly we are going to discuss about sodium potassium pump, calcium pump and hydrogen pump. So in the next session, we will be discussing about secondary reactive transport and what are the features and the functions of secondary reactive transport. Then, thank you.